The next speaker is Kevin Saunders. Welcome back, Kevin.
Thank you.
The title of his presentation is Vaccination Elicits Antibodies Against the Glycan Shield of HIV-1.
So previous observations have shown that the HIV-1 envelope is the target of neutralizing antibodies. One immune evasion mechanism HIV-1 uses is to heavily glycosylate its protein. Shown here in this model of the trimer, you can see glycans in purple and white. This shield of host glycans functions to cover potential protein-dependent antibody epitopes. Furthermore, the glycan shield creates heterogeneity among ONV molecules. However, the human immune system has found ways to combat the glycan shield during natural infection. There are several families of broadly neutralizing antibodies, or BNAbs, known that bind the glycan shield alone or in combination with the surrounding protein. 3 of them are shown here on the left, with PGT135, shown here in blue, PGT128, shown here in tan, make both protein and glycan contacts. 2G12, shown here in green, makes contacts with only the glycan alone. Interestingly, all 3 of these antibodies make contacts with the asparagine N-linked glycan, shown here in red at position 332. These antibodies have substantial breadth and potency, which makes them of interest for vaccine design. However, despite tremendous efforts with ONV-based and glycoconjugate-based vaccines have yet to elicit these types of antibodies. So our study had a twofold purpose. First, to determine if HIV-1 ONV vaccination can elicit antibodies against the anti-ONV glycan-dependent antibodies. Second, can advances in glycan synthesis and sensitive B-cell technologies enable the detection of anti-ONV glycan antibodies after vaccination? In this non-human primate study, the vaccine regimen was a DNA prime and an adenovirus boost, followed by repetitive protein immunizations with a group N consists of. We chose this Con-S ONV as our immunogen since Heather Desire at the University of Kansas has defined the glycosylation profile and has found that Con-S has a high abundance of oligomannose, which is the target of broadly neutralizing antibodies, around the V3 or N332 site. To determine the immunogenicity of this vaccine regimen, we use plasma blocking assays to block 2G12 binding, as well as glycan arrays and antigen-specific B-cell sorting. So first, to define the specificity of the antibody response, we examined if plasma antibodies could block 2G12 binding to ONV. Shown in red is blocking obtained by macaque plasma from animals mock immunized, and in blue are animals immunized with Con-S. We compared blocking at week 2 to week 199, which is after the 14th protein boost, since week 0 samples were not available for this assay. We observed blocking of 2G12 by most of the animals immunized with Con-S. Although not shown here, we also observed the similar blocking with against PGT128. However, the plasma from these animals show tier 1 neutralization, but they do not neutralize tier 2 viruses in the TZMBL assay. The 2G12 blocking suggested that glycan antibodies may be present, so we sought more direct evidence for direct glycan binding antibodies. We asked the question, can we use glycan arrays to detect anti-glycan antibodies in purified plasma IgG from Con-S immunized rhesus macaques? These arrays were coated with oligomannose glycans that comprise 96% of the glycan shield, shown here across the top. Also, the N-acetylglucosamine, or GLICNAC2, was included to determine mannose specificity, so this one here. We first tested PGT128 as a positive control antibody and found that it bound in a pattern identical to what has been previously published, so shown here. And then we also tested 19B as a negative control since we know that it is not glycan dependent, and it showed no binding. We next sought to isolate glycan dependent antibodies, or we next sought to compare plasma IgG of macaque M636, which showed high PGT128 blocking at week 2 and week 199. We found substantially high binding by plasma IgG at week 199 compared to week 2. These antibodies appeared to be mannose-specific since they only showed low binding to the GLICNAC2 by itself. We next sought to isolate anti-glycan antibodies directed against the BNAb epitope at position 332 from Con-S immunized rhesus macaques. We chose an approach of epitope-specific B-cell sorting to isolate these antibodies. As a positive selection antigen, we used ConC GP120 that has both the glycan at position 332 and 301. And that's what's depicted here on the HIV-1 outer domain, shown in green, and the glycan at 332, shown here in cyan, and 301 in orange. To gate out antibodies not to this site, we use a ConC GP120 with an N332 substitution, alanine substitution shown here, that has eliminated the glycan at 332. We tested these antigens against PGT128 and confirmed that they did indeed knock out PGT128 binding. We labeled these antigens with fluorophores and sorted memory B cells from the PBMC of M636. The events shown here in red are sorted memory B cells that had a PGT128-like binding, that they could indeed bind the wild-type ONV, but did not bind when there was an N332A mutation. These antibodies were recovered by RT-PCR and expressed by transient transfection. To determine if these antibodies bound in a 332-dependent manner, we screened a glycopeptide that has the glycans at position 301 and 332. We found that there were 3 antibodies from macaque M636 that bound in a glycan-dependent manner. So shown here in blue is binding to the glycopeptide when MAN9 glycan is added to the peptide, and you can see there's no binding to the same peptide when the mannose is not present. We have also performed similar experiments with a second macaque, M172, and this macaque showed a similar binding pattern for 2 different antibodies that we screened. And so you again see in blue it binds to the glycopeptide but not the aglycone version. This finding at the monoclonal level gave us confidence that our glycan-dependent antibodies could be elicited by vaccination. So we next asked the question, are glycan-dependent antibodies elicited by other vaccine immunogens? Due to its use in current and past human vaccine trials, we analyzed the oligomannose glycan array, IgG from a macaque immunized 2 times with AE8244 GP120, which David Easterhoff just spoke about. We purified IgG from plasma isolated before vaccination, as well as 2 weeks after the second AE8244 immunization. When we analyzed this by glycan array, we detected high binding by the purified IgG to MAN6, MAN7, and to MAN8 by glycan array, which provided another example of vaccine-induced anti-glycan antibodies in non-human primates. We hypothesized that this finding may translate to human vaccination. Therefore, we asked the question, are glycan-dependent antibodies elicited by AE8244 immunization in humans? So David Easterhoff in our group has isolated several monoclonal antibodies from RV305 participants, which he just spoke about. He kindly provided 2 different antibodies whose binding is sensitive to deglycosylation of ONV. I tested these 2 antibodies by glycan array, and shown here is the binding data for these 2 antibodies. As you see in blue, 1 antibody showed no reactivity to glycan. However, we did see reactivity by the second antibody shown in green to MAN5, MAN6, and MAN7, although there was some binding to glycan-2. So our ongoing studies, we are still in the process of producing large quantities of the 5 macaque monoclonal antibodies so that we can test their neutralization activity, their ability to capture virus, and to do structural studies if warranted. We are also interested in finding additional vaccinated macaques that have made this glycan-dependent response, so we're also performing large-scale screens of multiple vaccinated macaques. And then lastly, we are following up on the observation with the 1 RV305 antibody by examining a panel of RV305 antibodies from David Easterhoff in several different glycan-binding assays. So in summary, we have found HIV-ONV vaccination elicits antibodies that block glycan-dependent antibodies. Using glycan arrays, we have found plasma IgG in vaccinated macaques was capable of binding directly to oligomannose. We have isolated 5 monoclonal antibodies that bind to a V3 peptide mimic of the PGT128 epitope in a glycan-dependent manner. And lastly, we have found antibody responses against glycans in A244 GP120 vaccinated macaques and humans. So in conclusion, similar to natural infection, the glycans on existing immunogens near the N332 site of vulnerability are immunogenic. But since the plasma of ConS and AE8244 vaccinated recipients lacks neutralization breadth, if glycan-dependent BNAbs are indeed present, they are still a small subdominant minority of the glycan and the anti-glycan response. And so here's a list of the collaborators who have worked on this project and also our funding sources. And I'll take questions if time permits.
Any questions for Kevin?
You may have said this, and I missed it, but is there any cross-reactivity for these glycan-specific antibodies to non-HIV proteins that are glycosylated?
So that's one thing that we're actually planning to test. So it's a question that we've gotten before, specifically from Joe Sadrowski, who seems to ask that at each of our presentations. So it is something that we plan to test, but we don't know yet. We have not measured them. So right now they're still in the production process. So we don't have the large purified quantities of the antibody to be able to do neutralization yet. But we do plan to measure them.
Are the antibodies all in the same subclass?
So they're actually no. So we isolated 3, I think, that are IgM, and then the other 2 are IgG. I believe they're both IG2.
Just to follow up on Sudha's question, has anyone looked at the cellular reactivity of these types of antibodies, cross-reactive species themselves?
So it's similar to like a Hep2 staining. So no, we haven't done that, but we could look at that.